So in this video, we're going to learn about a type of synaptic learning referred to as long-term potentiation. What it is it's a way that we store memories. So for example, when you come to lecture and you listen to me talk, what'll happen is you are storing that memory in a part of the limbic system called the hippocampus. That is a short-term memory. Okay, and it can last from seconds to hours. So when you're learning something new, again, all that information first is going to the hippocampus, and it's being stored there temporarily. Okay. Again, it does last for seconds to hours, and it only has a certain amount of space. So if a lecture lasts too long, what'll happen is usually it's about an hour to an hour, 15 minutes. If it goes any longer to that, as you're paying attention, it's like the hippocampus is like a little person in there with a whiteboard, and they're writing the information down, but then they run out of space on the whiteboard. Okay. And so, in order to keep recording the information they're learning, they go to the beginning and they start erasing what was there. That's why when things go too long, you might forget something in the beginning because your hippocampus just ran out of space. But if that doesn't happen, you went to a lecture, you watch one of my lecture videos, they're about 40 minutes. I'm trying to get them all about there, less than an hour. Then you have this information, but here it says in your short-term memory in the hippocampus, it lasts for seconds to hours. What you need to do now is think about that information, right? This is what happens when you read over your notes, you read the textbook, but you have to do this within a few hours after you first learn the information. Okay, this is a key to help you do better in school, right? We all have to learn to do this. So if you just go back and you look at your notes, you don't have to do a long study session, but you just read them over quickly. What you're going to do is stimulate your long-term memory. Okay, long-term memory gets retained for days to years. Okay? And what you're actually doing is you're stimulating a synapse over and over again, which makes that postsynaptic neuron more sensitive so that the next time you think about that thing, whatever it is, homeostasis, those you will recall it because those neurons are more sensitive and will fire, and that's where the memory is stored. Okay. So what we're doing is we're consolidating the short-term memory into long-term memory. Consolidation is the process of transferring and fixing short-term memory traces into long-term memory stores. Okay, so how do we do that? This is an example of what you see with a presynaptic neuron. This is where the information would be coming in your hippocampus, and then you're going to store it into your long-term memory. And oftentimes that's areas in the frontal lobe. Okay. So there are several things you need to know about. We're going to go over the neurotransmitter that's being used, right? How it gets released. There's going to be two types of receptors on the postsynaptic membrane. Okay, so you need to know the two types of receptors. You have to know the action of those receptors. So one's going to be a chemically gated sodium channel. The other one is going to be a chemically gated calcium channel, but it's also going to have a gate in the form of magnesium. Okay? Once these are stimulated to open, then we're going to look at the sequence of events that occurs inside of the postsynaptic cell. And much of the sequence of events inside the postsynaptic cell, what it does is it causes an action on the presynaptic cell. One, to make it release more neurotransmitter, and within its own postsynaptic cell, it's going to make the dendrites and cell body more sensitive by inserting more receptors. Okay, here we go. The one nice thing that I did, I hope it's nice, is the next PowerPoint slide, I listed out And I even put in red some of the important terms that you learned about in the last unit. We're applying them in this unit. So I think what I'm going to do is I'm going to flip back and forth on this page to this one so that you can see the picture and see the words. Okay. So the neurotransmitter the presynaptic cell has stored in the vesicles is glutamate. Okay. So in the process, when you're thinking about this, what you're doing is the presynaptic cell is sending down an action potential, right? There's going to be voltage gated calcium channels here so that we can stimulate the release of the neurotransmitter from the vesicles. Okay. So you learned that last unit. So that's what happens. Glutamate gets released. Don't worry about this calcium. It's something different. It's something else that's going on, okay? Glutamate gets released. So that's what we see here. Whoops, on here. Number one, glutamate is released and it binds to the receptors that are on the postsynaptic membrane here. So you have to remember these two types of receptors. One is called an AMPA receptor, and another one is called an NMDA receptor. Glutamate binds to each of those receptors. There you can see the little pink ball on each one. Let's follow AMPA first. So AMPA receptor is also a chemically gated sodium channel. So when glutamate binds, AMPA opens, sodium comes in, right? Sodium's just floating around here in the interstitial fluid. So now that the channel is open, sodium comes in and we get depolarization. Okay, let's see what it says here. Right there, AMPA's channel's open, sodium diffuses in, it produces an EPSP. That's what that depolarization is. Now that EPSP is important in activating the other receptor, the NMDA receptor. Okay, not only do you need glutamate to bind to it, there you see there, but it's also clogged with a magnesium ion. Magnesium is Mg2+. It's clogged with a magnesium ion. So the depolarization or the EPSP that resulted as from opening of the AMPA receptor and channel, that pops the magnesium out. And your AMDA receptor is also a calcium channel. So that's going to allow calcium to enter. What does this say? So again, our glutamate bound to the AMDA and the AMPA receptors. On the AMPA, the channel opens, sodium diffuses in. It produced an EPSP. That EPSP clears the magnesium from the NMDA channel. So with glutamate bound and now the AMDA channel is cleared of magnesium, calcium flows in through those NMDA receptors. Now calcium is inside the postsynaptic cell. Now what's its job? It's going to do two things. Okay, we're going to see it activate a second messenger pathway. Remember we learned about those. And it's going to do two things. It's going to cause the insertion of more AMPA receptors, which increases glutamate sensitivity. There's going to be more receptors for glutamate to bind to, right? Which is going to make stronger EPSPs, okay? Which will open more channels, NMDA channels. Okay? It's also going to trigger the release of a paracrine. And that paracrine is in the form of nitric oxide. This paracrine is going to travel from the postsynaptic cell up to the presynaptic cell. Okay? And that's going to stimulate more glutamate. If you're going to have more receptors, you should have more neurotransmitter. Okay, let's see that happen. Okay, so we've got our NMDA receptor open now, or channel open. Calcium came in. Calcium stimulates a second messenger. One of the things that second messenger does is activates a protein kinase which causes the insertion of more AMPA receptors. So the cell body and the dendrites of this postsynaptic neuron just start inserting a whole bunch of AMPA receptors. Okay, so more sodium can come in and you'll get more depolarization, which will open more NMDA receptors. Okay, well, if you have more AMPA receptors now, and we have the ability to unclog more NMDA receptors, we should have more glutamate to bind to more of these receptors. So that's the other pathway. When calcium comes in, that second messenger also stimulates a second protein kinase. Remember, that's an enzyme. That causes the release of nitric oxide, which acts as a paracrine. So it's a paracrine messenger because it's going from the postsynaptic cell to the presynaptic cell. Okay, just through the interstitial fluid. It's not a neurotransmitter because neurotransmitters always go from the presynaptic cell to the postsynaptic cell. Nitric oxide is not a neurotransmitter. It is a paracrine, and we're going to see it later in the semester in another area too. Okay, so acting as a paracrine, nitric oxide stimulates the release of more glutamate. Now you have more receptors, so you have more glutamate to bind to it. The effect is you've made the postsynaptic neuron a lot more sensitive. So the next time you think about homeostasis or you think about this process, those postsynaptic cells are going to be a lot more sensitive, and that's going to be the easier recall of the memory. But if you don't think about it first right away, you've got to look at it after you learn it the first time. Go back and look at it within 20 minutes to an hour. That's you consolidating it to long-term memory. That's you doing this process. So use this, okay, these steps to help you understand this. It's also stepped out in your book. The other thing, calcium causes the growth of more dendritic spines. It just causes the postsynaptic cell to get more dendrites. So there's more room for receptors and things like that. It's a pretty cool process. I think it's pretty cool learning that way. Okay? So make sure you know this. I said this like 5 times now. Make sure you know this. All right, that you can step it through and explain exactly what happens in long-term potentiation. It's just the action of a synapse, which you've learned already, but now we're getting more specific with certain neurotransmitters and certain receptors and certain responses inside of the postsynaptic cell. The last thing I just want to say, in the hippocampus, neurogenesis, creating nerves, new nerve cells in the hippocampus. So again, that's really important for learning and memory, because the more you do this, then not only will there more dendrites and things develop, but also more neurons, postsynaptic neurons will develop, right? So that's what helps you remember things easily, okay? It also contains neural stem cells that continually produce these new neurons. The opposite can happen, though. If you're very stressed or even have depression, that can impede learning because your hippocampus actually shrinks. Stress and depression can cause the neurons to degenerate in the hippocampus. So you have less neurons, and it's going to be harder to consolidate, or harder to actually store a memory, right? Once you relieve the stress, or you deal somehow with the depression, it could be antidepressants or other means, you're in luck. The neurons come back. They come back to normal size, and your memory can come back. So did you ever, if you were so stressed out and you lost something, your keys, and you have no recall of where you put them, you go and relax, you calm down a bit, and all of a sudden you remember where they are. That's the hippocampus. So I remember that happening to me a lot when I was in college. So you got to learn how to manage the stress as well. Okay, thanks everybody.